All right. Very nice to see so many of you here. Welcome to the Bolin Center seminar. My name is Nina Kirchner. I'm one of the two directors of the Bolin Center for Climate Research. And I will give you a few practical informations on the seminar series in general and on some practicalities here. This seminar series is aimed to highlight research that is done in one of our research areas and to make it accessible to a broader audience. And we have 8 research areas in total in the Bolin Center, so the aim is to make your particular research understandable for all the non-specialists that are not experts on this topic. We've had very nice experiences so far with these seminars. This one is particularly well attended, and we also have people watching the stream, and this is why I talk into this microphone. The speaker then in a second will not have this microphone, but also later for the discussion you will have to use it, and it's not for you to hear me better, but it is for those who listen in the streaming session. So this is why we use this microphone here. We also have, for all those that are here, fika afterwards. This will not go to the poor people watching us. They can enjoy something else, but the fika is for those who are here. I will now hand over to Kalle Gotthard, who will introduce the speaker a little bit, and then it's time for today's speaker to take over. Varsågod, Kalle.
Okay, thank you. So it's really nice to see many of you here. This is then the research area 8, biodiversity and climate. This is our contribution to the seminar series. And it's super nice to be able to host this seminar with Albert Phillimore from University of Edinburgh. And Ali, he did his PhD in Imperial College, where he also stayed for a postdoc, and then he went directly as a research fellow to Edinburgh, where he stayed and has been the last 8 years, you said, yeah. His early work were more in macroecology and evolution, but his present work is to a high extent questions about how climate change might affect the phenology of organisms, that is the seasonal timing of biological events. And he uses a combination of citizen science data and also field observations to explore these questions, and also I think particularly interesting how potential phenological mismatches between species that are interacting with each other may be affected by climate change. And with that, I'll give the floor to Ali, please.
Okay, thank you very much for the introduction, Carl, and the invitation to come here. It's a real pleasure to be here, and thank you all for coming along today. So you've done a lovely job of explaining what I'm going to be talking about today. I'm going to spend the first kind of probably 2/3 of the talk discussing the insights we can make from citizen science observations, and then maybe about the last third talking about some of the field work we've been doing on this transect in Scotland. Well, for those of you who aren't working on phenology and aren't in this field, phenology is this timing of recurrent life history events. So for instance, the flowering times of plants, the emergence times, flight times of butterflies, breeding times of birds, frogs, things like this. And it's become a real hotbed for ecological and evolutionary research in recent years, really mainly due to the effects of climate change. So particularly at high latitudes, we find that spring temperatures seem to have a pronounced effect on phenology. We'll all have noticed that spring events are earlier in warmer years, and some of the best evidence of biotic impacts of climate change come from phenological time series. We know that phenology matters, timing matters, because it influences the abiotic environment an organism is in. So just imagine a frog that spawns too early, it may well be exposed to late frosts. And timing also matters for the interactions that are available with other species. So how much resources are available to a consumer, or how much opportunity to meet with mates are there. And because of this then, because of these two things of timing matters, then timing will impact, or we think timing will often impact fitness measures, so reproduction, survival, and it may also impact on population growth rates. So this is somewhere where if phenology is changing with climate change, timing matters, and timing impacts on fitness. This is one of those locations where we might find impacts of climate change on population growth and on evolution. So as I said, phenology of many different organisms at higher latitudes is getting earlier. And this is just some examples from my own group's work, taking citizen science observations and kind of examining how much different species can, or different groups of organisms can shift their timing by. So we find that trees in the UK are able to come into leaf round about 7 days earlier for every degree that it's warmer. A similar amount of shift we see in the flowering times of plants, so around about 6 days per degree. We also see a shift of around about 6.5 days per degree in butterfly flight times, and around about 4 days per degree in the egg laying dates of birds. So there seems to be some sort of ability to shift timing. And the main questions that that my research group are exploring and I'm going to talk about today then is, are these shifts enough to track changes in optimum timing, and are they enough to maintain interactions with other species, particularly trophic interactions, so feeding interactions? But many of our greatest insights into kind of phenology and the timing events come from these really long-term time series. And I'm not going to take that perspective today. Instead, I'm going to take a spatial perspective. And I've got two main motivations for doing this. So one is that we can potentially use space as a substitute for time. So there we have a temperature time series on the left. Over time, you can see the anomalies in temperature, but we also know that we have pronounced gradients in temperature in space as well. So perhaps we could explore how organisms respond to that spatial variation in temperature and use that as a proxy for how they might respond over time. The obvious benefit being it's much easier to study, to get a really good data set over space than it is over time. So to give you a kind of cartoon example of how that might be informative, imagine we had two populations of this bird that breed at different times in the north of Britain to the south. So day 100 in the south, day 120 in the north. If we say that the temperature difference is about 2 degrees C, now one scenario might be that this entire difference in laying date could be just driven by phenotypic plasticity. That's the flexible ability of an individual to have a different phenotype, so a different laying date at different temperatures. Now, if that was the case, if temperatures were to rise by 2 degrees over time, then we really would, on that basis, we'd predict the bird should be just fine. They'll just shift their laying dates forward. The alternative is that difference in laying date is all due to genetic adaptation, local adaptation, so a genetic divergence. If that was the case, then if temperatures were to rise, we would predict that the birds would be under selection for earlier laying dates, and that selection might come at the cost. So we have quite different predictions of how the birds would respond and be affected under those two different scenarios. The second motivation for taking a spatial perspective is that, as I said, lots of our best data sets on phenology are these really long-term time series, like Hoogvalu in the Netherlands, looking at great tits, and also White and Woods in Oxford. But they're often conducted in kind of a single type of habitat. And it's hard to know whether these single site studies, do they generalize in space? So by taking a spatial perspective, we can ask, how do phenological patterns that we observe in a single site generalize to different habitats, different locations? Okay, so this first part of the talk, I'm going to focus on what we can learn from citizen science. So I need to kind of give a huge thanks at this stage for the enormous efforts of citizen science or amateur naturalists contributing observations of phenology. So I know you also have similar schemes in Sweden, but in the UK, these three schemes are the UK Butterfly Monitoring Scheme, Nature's Calendar, and the British Trust for Ornithology Nest Record Scheme. All of these schemes have tens of thousands, actually millions of observations of phenological events collected across many years and many locations. So one of the kind of real attractions of working with these data is just the huge spatial and temporal scale of these data sets. Now, in that cartoon example I showed you a couple of slides ago, I talked about the interest in knowing whether populations are show local adaptation, so genetic divergence, and in response to temperature in space. And our classic methods in evolutionary biology for testing for local adaptation involve reciprocal transplant experiments where we move populations, we examine their fitness in their home environments and away environments and test whether they are fittest in their home environments. Now, this is a really nice approach, but it has logistical challenges. That means that most of our insights into local adaptation come from groups that you can apply these experiments to easily. So we have good insights into plants and some invertebrates, but next to no insights into the local adaptation of other organisms such as birds or mammals. So myself and Jared Hadfield and Owen Jones started thinking, well, could we develop a statistical approach to try and get at a similar thing, but only using citizen science data? And so I'm going to present that now, but acknowledging that there are a lot of caveats with this approach. It's not as good as a reciprocal transplant experiment, but it's something you could apply as a first test or where reciprocal transplant experiments aren't possible. Okay, so the premise of this approach is that if you look at the single population over time, and you know what the temperature Q, so the time during which a population responds to temperature, you could see what's the relationship between phenology and temperature over time. And if we're looking at so there we think this relationship, it's also known as a reaction norm, this should be due to phenotypic plasticity, so this flexibility, and genetic change, adaptation. But if we're looking at a very short period of time, then most of that change, I'd argue, will be due to phenotypic plasticity. Now let's imagine that you can imagine that the same temperature Q is important for different populations as well. So you can instead look at the relationship between phenology and temperature across different populations, so a spatial slope. And that slope should be due to phenotypic plasticity plus some contribution of local adaptation. And if the populations have been in situ for many generations, then I'd argue that local adaptation has had plenty of opportunity to contribute. Now, if we assume that plasticity is a constant, you'll see that the difference between that spatial slope and the temporal slope gives us kind of a statistical inference of what temperature-mediated local adaptation of phenology exists. So that's the premise of our statistical approach. And here I'm going to give you an example of where we've applied this to a couple of data sets. So this work was conducted with Sandra Stalhansky, who I think will be familiar to many of you here, when she was working on her master's at Imperial College. And this was using nature's calendar observations of the first flight dates of orange-tip butterflies, the first flowering dates of these two host plants in the UK, Cardamine pratensis, so cuckoo flower, and Aliara petiolata, garlic mustard. And the first step we had was to identify when these species were responsive to temperature. So what I've shown here is just what's known as a sliding window approach, where we just try average temperatures over lots of different time periods and ask which time period is most predictive, over which time period is temperature most predictive of phenology. And you can see that there is a period in early spring in all three cases, highlighted in orange and green, when the temperature is most predictive, but you can also see there's some uncertainty. So we don't know exactly when these time windows are. If we take these time windows, we can then use a statistical approach to estimate separately the slope of phenology on temperature over time, shown with gray lines here, and over space, shown with the black line here. And so if we take the two flowers first, what we notice then is that the slope over time, a slope of around about 6.78 days here and 5.63 days for the cuckoo flower, those slopes, so this is what we infer is plasticity, and this slope is very, very similar to the spatial slope. So there's no difference between the spatial and temporal slope. Therefore, we've got no evidence for any local adaptation of these flowering plants. If instead we look at the orange-tip butterfly, we see that over time, the slope is a tiny bit steeper, so minus 7.74 days per degree. And over space, here, the black line, this slope's quite a bit shallower. And in this situation, the slopes are different over time and space, and potentially we have evidence for local adaptation here. And I'll come on to some caveats in the next slide. But it's intriguing here that the local adaptation is kind of counter-gradient. It's leading to the populations being more similar at different temperatures in their phenology than you'd expect if they were just responding plastically. So as I say, one of the strengths of this statistical approach is it's quite easy to apply to these large existing data sets. And David Roy and Tom Oliver applied this approach to 31 species of butterflies in the UK. And so here we have the spatial slope in each case versus the temporal slope estimated for each of these species, with a dot for each species. Now what you can see is in every case, the temporal slope is negative. So all of these butterflies seem to be highly plastic. Their peak flight period is around about 6 or 7 days earlier if a year is 1 degree warmer. But what you'll also notice is that most of the dots fall below the line, meaning that these butterflies are responding less to temperature in space than they do over time. And in 14 out of the 31 cases, the slope is significantly shallower over space than over time. So one interpretation of this is that there's this kind of consistent counter-gradient variation in the butterflies. And here the idea is that if they just responded with plasticity, the phenology would be down here, and adaptation, evolution has taken them back up here to making them much less variable in space in their phenology than we would expect through plasticity alone. So one interpretation of this is that we've got evidence for really persistent local adaptation in many butterfly species. But a more cynical perspective might be that you generally don't find such consistent results for any other reason than statistical artifacts. And so it's quite possible that there is another variable that our models are missing that co-varies differently with temperature over space and over time, and with phenology over space and time. And some of Carl's group's work suggests that one of the candidates for that might be some sort of chilling requirement that we're missing from our models. So I think these models are, this kind of macroecological approach is a really interesting way of raising questions that require confirmation in the future, but they're not, they don't give you robust, conclusive answers on their own. Okay, so what I'm going to move on to now then is some kind of more recent advances to this statistical approach that have arisen from some theory developed by my colleague Jared Hadfield. So here, imagine these grids represent different populations that have different mean temperatures in space. And Jared's theory paper argued, well, in this situation where there's no kind of decline in temperature, then individuals will migrate between adjacent cells, and it means that they'll never, at any single one of these cells, you'll never find a population actually at its optimum phenotype for that temperature, because individuals bringing maladaptive phenotypes will always be coming in. So populations won't quite manage to track the optimum if temperatures kind of spatially heterogeneous. Alternatively, if there's a temperature climb, as shown here, then it is possible for adaptation to track the optimum along that climb. And this is because if you imagine that populations are kind of in the same numbers everywhere, then individuals coming from warmer temperatures into a cell will be counterbalanced by individuals coming from colder cells into a cell, so that along that particular climb, populations should be able to track the optimum, at least in theory. So then taking this back to kind of the real world and away from theory, we know that there are pronounced spatial temperature climbs. So in the UK, the south is considerably warmer than the north in particular. And Jared pointed out then that if you look at the relationship of temperature regressed on latitude and phenology regressed on latitude, then the ratio of those slopes, so the phenological climb divided by the temperature climb, tells you how much, if you assume that that populations are able to track the optimum along latitude, it tells you how much does the optimum phenology change for a 1 degree change in temperature. So we were really excited about the potential to estimate this thing, this thermal sensitivity of the optimum. And here I'm going to show a really horrible looking equation, but it's a really cool equation actually. This is from a paper by Luis Miguel Ruslandi and Georgina Mace, where they set out to try and develop a model that we could parameterize with real world data to try and predict the maximum rate of environmental change, or maximum rate of temperature change that a population can persist in. And one of the things they point out, so this little b is how much plasticity does the population have with respect to temperature. And they point out that this big B, which the thing I've just been talking about, this environmental sensitivity of the optimum, this is really pivotal to estimating how a population might cope with climate change. And yet this has been really, really hard to estimate. You need many years of data. You need to track fitness in many different years. And we have, the reason we're excited about our approach is we have this method for trying to estimate this capital B using citizen science data alone. So the key here is then if the difference between the environmental sensitivity of the optimum and plasticity, where that absolute difference is small, those are the populations that should cope with climate change fine. Where that difference is large, there's a much greater amount for evolution to make up. So our first application of this kind of extended approach then was to data from the British Trust for Ornithology on the first egg dates of birds. We focused on 4 bird species, the blue tit, great tit, chaffinch and pied flycatcher, for which there are huge amounts of data and they're mainly single brooded species in the UK. So to give you an idea of the number of observations, for blue tits, there were 35,000 observations of first egg date across the UK, going back to the 1960s. And what this shows here then is that all of these species are shifting their phenology to be earlier in the year over time, and there are also quite pronounced spatial gradients in their average egg laying date across the UK. So the first thing we had to do then is to estimate how plastic each of these populations are. And we find for all 4 species, they show significant plasticity. So these bars are 95% credible intervals from a Bayesian statistical analysis. So all of these birds lay their eggs around about 4 days earlier if a year is 1 degree warmer. So although we've estimated plasticity just at the population level, our estimates are very consistent with studies that have followed individuals. So studies from the likes of White and Woods group. One possibility is that different populations would have different plasticity. Each one of these blue lines corresponds to a different kind of grid cell across the UK, so a different part of the UK. And you can see, I think, that these plastic responses of different populations are remarkably similar, suggesting that there really isn't much variation in the plasticity across the UK. So we then wanted to know, is there any local adaptation in the UK, and what is this latitudinal slope ratio, this environmental sensitivity of the optimum? So this is the estimate we get for that. What you'll notice if you compare this environmental sensitivity of the optimum and the plastic slope is they're remarkably similar. So this is informative in 2 ways. First of all, we don't have any evidence for local adaptation of these birds across the entirety of the UK alongside latitude. And it also means that it looks like these birds will be pretty bulletproof under climate change, at least if these reaction norms remain linear for some time, because the plasticity does a really good job of tracking this environmental sensitivity of the optimum. And we extended these ideas a little bit further. So we, remember before I said you could look at the regression of phenology on latitude and temperature on latitude and that ratio. Well, you can do the same with year as well. So you can ask how much has phenology changed over time? How much has temperature changed over time? So what's that ratio? And you can ask whether that year slope ratio, is that the same as the plastic response, or is it greater or smaller than the plastic response? So this, on the y-axis here, we have this year slope ratio minus plasticity. So if plasticity was kind of tracking this temporal change perfectly, we'd expect those bars all to fall on 0. And we can see that in 3 cases, the bar 95% credible interval does include 0. But in one, the pied flycatcher, it looks like the birds have shifted their laying date more over time than expected solely on the basis of plasticity. So one possibility is this reflects some evidence for adaptive microevolution, so that they have evolved to have earlier laying dates. So I think it would be right to be skeptical of that conclusion, but some evidence that that might be true is the recent work coming out of Barbara Helm's research group in Groningen. They were using a common garden experiment through time with pied flycatchers, and they found evidence for adaptive microevolution in pied flycatchers. So perhaps we are detecting a similar pattern that they've identified in Germany. Of course, as with the analysis of the butterflies, we have to have the caveat that these approaches are based on kind of regressions, or based on 2 regressions, one a regression between year and temperature, and another regression between year and phenology. And of course, any correlative study like this can always be kind of undone by a 3rd variable that we've missed. So we have to kind of be aware of these caveats. Okay, I said at the outset that one of the kind of motivations of the work we're doing is to understand how phenological interactions between species might be maintained or might be disrupted with climate change. And citizen science data gives us opportunities to try and reconstruct some of these phenological interactions. Not many, but some. So as I said, with the orange-tipped butterfly and its 2 host plants, we have observations in the UK of all of these species. And we can ask, if we look at the flowering times of the plants versus the first observations of the orange-tipped butterfly, how much of the flowering plants are available for these butterflies to lay their eggs on. And what we find then across the UK is that in the north, there's greater synchrony with the cuckoo